Okay, we are on to chapter 2. Chapter 2 is where we really start to get into the details of talking about disease, which doesn't always relate to microbiology, right? We have many diseases that aren't caused by microorganisms. We have things like diabetes and cancer and stuff like this. But a lot of what we call infectious diseases, things that can be transmitted, are caused by microbes. So these kind of go hand in hand, but there are some really critical definitions in this section that help us describe what we're talking about in terms of things like, is an infection the same as a disease? No, they're actually not. And we'll talk about a few specifics, and we'll start this section with a case study. So let's look at our objectives here. So we want to talk about the relationships between things like the host, which is the generalized term that we use for whatever's being infected, right? It's hosting the other organism. Its microbiome. So we'll really define that very, very clearly. I've talked about it several times, but I'll be very specific. And what we call pathogens. So not all microbes are pathogens, and this is a very specific term, and I'll go through the definition in a little bit. We'll then apply kind of what we call the basic concepts of pathogenesis to kind of a specific infection, what makes a microbe a pathogen, and talk about how infectious diseases impact communities, how things get spread through them, and how communities themselves kind of influence pathogens in the other direction. All right, so this is a less fun case study, but I think an important one. So we have Brandon. He's a 30-year-old stockbroker living in Chicago, and he goes into the physician's office. When the nurse asks him why he's there, he's a little embarrassed, but he says he needs to talk to the physician about a health problem. Warning, here comes the penis with the lesion on it. So obviously he has some sort of sexually transmitted infection, which unfortunately is more common than we like to think of, and can be very embarrassing for people to deal with, but in a lot of cases often easily treated when identified and caught early. So the physician examines him and notices the small lesion that is leaking clear fluid from the end of the penis. So he says it doesn't really hurt. It doesn't look great, but it's very clear what it is. The physician asks him some questions, and Brandon ends up saying that he has been sexually active with more than one partner. So that's often a higher risk scenario because there's the risk of sexually transmitted infections there. So we want to then identify what is causing the infection here. And to do that, the physician is going to take a sample of that fluid and send it to the clinical microbiology lab, where the first thing they're going to do is actually look at it under a microscope, which is really simple and really easy. And they're going to see that there are these kind of corkscrew-shaped bacteria that are swimming around in the sample. That's a really clear indication that this is caused by a very well-known microorganism. It's caused by Treponema pallidum, which causes the disease we call syphilis. The French pox, the English pox, the Spanish pox, every culture has a different name for it. They usually blame their neighbors for this. This disease, if you're interested in the history of syphilis, there's a great story about it in the textbook in the intro to this chapter about how it spread from the New World. As explorers were coming back to Europe, they brought it with them, and as they sailors moved around between partners, it spread. And syphilis, actually, if untreated, is very dangerous. There are a lot of places that it can go besides the urogenital tract. It can get into the brain and cause all kinds of issues there and ultimately lead to death. Luckily, we live in an age where we have antibiotics, and antibiotics are chemicals that destroy bacteria and not humans. So the physician prescribes him long-acting penicillin, gives him a shot, and that leads to the lesion disappearing over several days, and he's going to have to contact his sexual partners to make them aware of this. Either he spread it to them or they spread it to him, and try to stop that chain of infection. So, that's like a broad example of microbiology influencing our life, right? Hopefully you never have to experience that, right? But it's not necessarily important to know the details to understand what's going on here, right? You understand how it's spreading and things, but when we know the details about how pathogens work, it really helps us demystify it. It's less of a black box that we have to go to someone to fix. We can start to understand how things spread and how we can protect ourselves and how we can identify infections and take precautions and even realize when we need to go get something treated. So understanding the details is really important for medical professionals, but the general public, right? It leads to less fear from these things and understanding how we should deal with them and what are reasonable precautions that we should take. So in chapter 2.1, we're going to talk a lot about what is normal microbes versus pathogen. I use normal in quotes, right? Because that's a tough definition, right? What's a normal microbe versus a different one? So we'll talk about the differences between our normal microbiota, things that live on and in us, versus pathogens, which are microbes that cause disease. We'll discuss the relationship between infection and disease, and then between what we call virulence and pathogenicity. And then we'll actually differentiate between some numbers. We want to quantify things a lot of times in science. Things like infectious dose versus lethal dose. That's how much of a microbe does it take to get infected, and then how much does it take to kill you, which can be important to know. And then talk about what really makes a successful pathogen, right? A successful pathogen is a very good pathogen. That's not good for us. That's good for the pathogen, right? Usually that's bad for us. So if we know what makes a good pathogen, we can attack those things or avoid those things and not die from infectious diseases, hopefully. Okay, so let's start with what is a pathogen. But let's start broader than that, right? Let's think about our human bodies and all over our body, right? On my hands, in my teeth, in my hair. There's microbes everywhere. That sounds gross, but it's normal, right? None of us are completely sterile. We all have some sort of microbes. And in fact, we probably have as many microbes in us as we have cells in our body. Some estimates put it even higher. There might be more microbes than there are human cells in your body. So you're mainly microbe, actually. So we call this the microbiota or the microbiome. That's the collection of all the microbes that are on and in the human body. So everything that's on you, everything that's inside of you normally, that's our microbiome. To do this, microbes have to colonize, and colonization refers to the ability of a microbe to stay attached to a body surface and then replicate there. That could be in the intestines, like these E. coli here. They might have some molecules on the outside that stick to things on the surface of our cells and allow them to stay there, right? But if they can't colonize, they're going to get excreted very quickly or washed off of us. So colonization allows microbes to stay in and on us. Okay, here's the critical term. Pathogen, right? I've talked about bacteria and microbes. Not all microbes are pathogens. In this case, a pathogen is any bacterium, virus, fungus, protozoa, or worm, which often get called helminths, that causes disease in humans. So here's some examples, right? We have athlete's foot, which is toe fungus, basically, or foot fungus, right? It is literally a fungus that grows on us, and it causes a disease. It causes painful irritation, burning, can cause nails to fall off, things like this. It's ultimately, not the most deadly pathogen, but it does cause a disease, right? It doesn't often kill us, but it is disease. Then we have another one here. Here's a worm that causes what we call elephantitis. This gets inside the body and causes this swelling to occur as it infects our tissues. So this leads to some interesting points. Not all pathogens, right, live in us. Some live on us, like the fungus here, but some live inside of us, and some even live inside of our cells, right? They get inside of the cell itself. So if we talk about pathogens, right, we have this term called pathogenicity that we can use to describe an organism. That's the ability of an organism to cause disease. How much disease can it cause? How bad is that? That's kind of what pathogenicity is. Now, in common usage, we have this term parasite. That's kind of like anything that lives on us, right? But in medical microbiology, parasites are a very specific type of organism. They're usually eukaryotic or protist microbes that cause infections. So think like Giardia, or the malaria-causing parasite, or this worm is a parasite. But we generally don't refer to, in medical microbiology, bacteria as parasites. So just be aware of that distinction. There's some distinction there in the medical term. Okay, we're going to continue with our description and definitions of words. I know it's a little boring, but it's actually really important. And you don't want to use the wrong term in a medical context because it has really specific meaning, and people will know if you know what you're talking about or don't by how you use these words. So it's really important to not only understanding things, but showing that you know things by using the correct terms. So let's use one that often gets misused. An infection. An infection occurs when a pathogen or parasite enters and begins to grow in or on the host. So you get a rhinovirus, which causes the common cold. Once it goes inside of you and starts to replicate, that is an infection. Most infections don't cause symptoms, or they go unnoticed. So symptoms are things that we can feel, see. We'll talk about signs and symptoms a little bit later, but they're outside things or things that we can feel that show that we have an infection. Most infections don't cause this though, right? A bacteria gets into us and starts to divide. Our immune system comes in and gets rid of it right away. You never know that happened, right? So you may have lots of infections going on right now, but your immune system, if you have a healthy immune system, is just dealing with them. Now, the term disease occurs when a patient develops symptoms. So things that we can see or that the patient can feel: pain, sweating, redness, pus, things like this, fever. Those would be symptoms, and that indicates that a disease is occurring. So you can have infections that don't cause disease. That's really important to know, right? Not all microbes are pathogens. Remember, a pathogen is a microbe that causes disease. We can classify a disease kind of based on its progression. Some diseases are what we call, they have an acute infection, which means symptoms develop and go away rapidly. Think the common cold, right? Couple of days. Then we have chronic infections where the disease, the symptoms develop gradually and take a long time to resolve. If you think back to chapter 1, we talked about that tuberculosis example where like half the school ended up being infected with it, but they didn't know because the infection developed so slowly. Tuberculosis is something that sticks around for a long time, years, and it takes a long time to grow and develop, and symptoms take quite a long time to develop. So this distinction, infection versus disease, is really critical. If there's one thing you take away from this, 1, know the definition of a pathogen, and 2, know the difference between infection and disease. Okay, so let's talk about pathogens. We have some pathogens that we call primary pathogens. These are very likely to cause disease after infection in a healthy host, right? A healthy person that gets infected with a primary pathogen is highly likely to develop some sort of disease from it. That means the microbe is going to rapidly reproduce and increase in number in there, and it's going to have moderate to high what we call virulence. I'll show you what this means in a moment. An opportunistic pathogen is something that's less likely to cause disease in a healthy host. We have all kinds of things like that, but an opportunistic pathogen might cause a disease in someone with a weakened immune system. Say somebody's infected with something, right? How about, let's take HIV as an example. HIV, primary pathogen, right? If you're exposed to HIV, very likely it's going to get in and cause disease, which it will lead to AIDS, which is the disease, right? The lowering of the immune system. Once your immune system starts getting destroyed by the HIV virus and you develop AIDS, you're open to opportunistic pathogens, other bacteria that healthy people normally just fight off and have no problem, are now very deadly and dangerous to people with weakened immune systems. So that would be an example of an opportunistic pathogen. We'll talk about some of these later in the course, things like Giardia or Toxoplasma. Can be very deadly to people with weakened immune systems. Now, some pathogens also have a different state. They can get into us and then go into what we call a latent state, or basically go dormant. Think like herpes viruses. So herpes, I know, has the sexual transmitted infection connotation to it, but there are lots of different herpes viruses, including cold sores. If you've ever had like a canker sore or cold sore, those can be caused by herpes viruses that actually go into our cells and go dormant, and they kind of stay around in our body. And then when some sort of event happens, like stress or things like this, they can actually reactivate. So how do we define pathogenicity? Well, we can do it in two ways. We can talk about what we call infectivity. This is how easily an organism causes disease. And then a really important one, which is called virulence, which is how severe the disease is. And we're going to have actual specific ways to measure this. So think about this. How do you measure how severe a disease is? Think about some ways. Well, that's really tough, right? Because it's kind of subjective. For virulence, unfortunately, we generally use death as our measure, right? Because that's very easy to observe. How severe a disease is, if it's you're dead or you're alive, that's really easy to see. Other things aren't there. So we use these for the worst of the worst diseases. Infectivity, this is a little bit easier, right? We can test people to see if they have the pathogen after they've been exposed to it. So let's look at some examples. Let's take everybody's favorite scary pathogen, probably the one of the scariest pathogens out there, Ebola. It is what we call highly virulent. So remember, that's how severe the disease is. It's highly virulent because the fatality rate is about 50%. If you are infected with Ebola, you have a 1 in 2 chance of dying from that. That's pretty crazy if you think about it, right? That's really scary. That's why this healthcare worker, here's the Ebola virus, this person is likely infected. That's why they're completely covered with protective gear, even goggles to prevent any fluids from getting in their eyes. Because once you get it, there's no cure for Ebola. It's a virus. We don't have very many antiviral drugs. And if you do get it, you have a 1 in 2 chance of dying. So that's scary. Highly virulent. Now let's take the other side of things, right? The common cold, our rhinoviruses, our stuffy nose cold. Well, it's really good at causing disease, right? But it almost never kills anyone. So we would call this highly infective. Remember, infectivity is how easily an organism can cause disease. So it's highly infective, but it has a very low virulence because it rarely kills people, right? So we can actually measure this, and scientists do experiments with this because we want to know how likely things are going to spread. So I'm going to start with what we call the infectious dose 50%. I'll get to the 50% in a moment, but the technical definition is the dose or amount of whatever, a microbe, needed to colonize 50% of an experimental group. So we're going to use an example of how many bacterial cells, or it could be viral particles, does it take until half of the sample group becomes infected. So I have 3 sample groups here, each with 6 individuals. Blue means they're not infected, and red means they got infected. So my 3 groups of 6. Okay, in the first group, I expose each patient to 1 bacterial cell. This is just a thought exercise, right? That would be very difficult to do to just get 1 bacteria, but imagine, right? I expose each of them to 1 of a bacteria, right? And none of them get infected. Okay, let's up it a bit. How about we take our next experimental group and expose them to 10 bacterial cells? All right, in this group, we had 1 that gets infected. So 1 out of 6, that's not half. Okay, my next group, I give each patient 100 bacterial cells. In this case, 50% of the group, 3 out of 6, got infected. So that's my infectious dose 50 level, right? That would be a really scary amount if it was just 100 bacterial cells that it took to get infected. That'd be very infectious. But in most cases, it's a much higher number than that. But this is an example of that, right? And this is really important to know because, we want to know how likely this is going to spread. The lower the infectious dose, the more likely it is to spread. So with only 100 bacterial cells, all right, that's very easy to spread versus something that takes 10,000, 100,000 cells, 1,000,000 to infect someone, right? That would be a low infectious infectivity. Okay, so then back to virulence, right? Describing the level of harm caused by a pathogen following infection. Well, to do this, we use what we call lethal dose 50 or LD50. This is the dose of bacteria or virus needed to kill 50% of an experimental group of animal hosts in this case. Okay, so let's look at this graph. This is a more realistic graph. On the left, on the y-axis here, we have what we call percent mortality. So remember, we have those groups, let's say of 10 individuals, right? Or in my case, 6. So I give one of those groups 100 organisms. This is our dose on the x-axis here, the number of organisms. Let's say it's bacteria. I give them 100 bacteria, 200 to another group, 300 to another group, 400, so on to individual groups. And then I look at the percent mortality. Mortality means death in each of those groups. Now let's look at the blue line, which is infectious agent 1. It's just one type of bacteria. Okay, at 100 of this bacteria, less than 25% die. Same thing with 200, 300, less than 25%. Okay, and then we go to like 350 here or around 400, and it jumps up real quickly to 50% die. Okay, so it took 400 organisms of agent 1 to kill 50% of a sample group. For agent 2, it takes a lot longer. It takes all the way up to 600 organisms to cause 50% death. So think about this. Which of these is more virulent? Agent 1 is more virulent because it takes less to kill 50%, right? You need less of it to kill, right? It's very deadly. Whereas Agent 2 took more to kill, so it's less virulent. Okay, so this is actually something that gets done as well. We can also use a similar kind of curve and setup for testing chemicals, right? We want to know how toxic chemicals are. But in this case, we're looking at our virulence level, right? We also want to describe what we call the host range, right? So the host is the thing that gets infected. The host range refers to the animals, which includes humans, right, that a pathogen can infect and produce disease in. And so this could be narrow, right, or broad. So we have some organisms that can infect only one type of host, one single species, and produce disease in there. That would be a narrow host range. In our example, we're going to talk about this Salmonella enterica, and we have this term serovar, which is kind of like, how do I describe this? It's like a specific strain, okay? So it's the same species, but it's got a few differences that make it only infect humans. This is Serovar typhi. We have a similar Salmonella enterica, Serovar typhimurium, that infects other animals and humans. So this one can infect many types of hosts. It has a broad host range, right? Why is this important? Why do we care about host range? Why do we care if something can infect animals? Well, one, farmers might care, right? Because if it infects their animals, it could kill them. Two, we care about it because we interact with animals. Let's think about our SARS-CoV-2, our COVID coronavirus. We think that came from interactions between humans and bats, right? Bats can get this coronavirus, and someone interacted with that bat and it transferred from it. This is what we call a zoonosis. We'll talk about it in a bit, but a lot of our new diseases may come from pathogens that have broad host ranges. So it's really critical to know this, right? Where it can spread from and what we should be aware of. Okay, so what happens when the pathogen meets the host? Well, it's got to do three things. The first thing, as we talked about, is it needs to attach, right? Otherwise, it's going to get washed off or excreted or whatever. So microbes often have special proteins on their surface that allows them to bind to what we call host cell receptors. These are like things on the outside of cells that usually get signals, right? But the microbes trick them into holding on, and they attach to our cells. Once they're attached, right, they can start to colonize. But when they do that, they have to start to avoid the immune system. Our immune systems are incredible. We're going to talk about them later in the course. They have so many ways of recognizing and fighting microbes, but microbes are just as clever. And pathogens, they find ways to evade and trick the immune system. There's all kinds of ways we'll talk about. They can live inside our cells. They can actually coat themselves in pieces of our cells and kind of hide and say, nope, I'm just a cell here. I'm not actually a pathogen, and the immune system can't find them. So there's all kinds of clever tricks that pathogens do, but they have to avoid the immune system. And then to grow and reproduce, they're going to need to steal nutrients from the host. They're going to need those to divide. And a big nutrient that they actually need is iron. So iron from the blood, sugars, things like this, proteins, carbohydrates, all the things that a growing bacterium needs, right? So these are the three things that a pathogen needs to do to live in a host and cause disease. Okay, that's a lot of terminology, but it's really critical to get that down because it's how we describe microbes. So, infection with a microbe doesn't always lead to disease. That's a really critical thing, right? We have pathogens. Those are disease-causing microbes. Primary pathogens, those are the bad ones, most likely to cause disease. And healthy individuals, opportunistic, likely to cause disease in immune-compromised host. Pathogenicity refers to mechanisms a pathogen uses to produce disease, and we can measure that, talk about how efficiently it can do that. Things like virulence, how severe the disease it causes is. Invasion, it's got to get inside. We have a term invasiveness there. Host range, what organisms can it affect? All the different animals or plants or whatever. And pathogens, they got to be able to attach to host cells, evade the immune system, at least temporarily, and obtain nutrients from the host. All right, that's it for 2.1.